let's review some components of the adaptive immune system. Our adaptive immune system provides the final line of defense against an infection. The adaptive immune system is activated after the start of this innate immune system. In fact, the innate immune system is required to activate adaptive immunity, and they crosstalk and work together. In adaptive immunity, we will target specific cells or organisms that our body recognizes as foreign. What's important and unique about the adaptive immune response is that it remembers these pathogens later, so that when we see it again, it already knows what to do and it can mount a heightened immune response. There are two arms of the adaptive immune system. The humoral immune system is made of our B cells and antibodies. The humoral immune system targets extracellular microbes and toxins, whereas cellular immunity is made up of T cells, and cell-mediated immunity attacks intracellular microbes, killing infected cells. Key to stimulation of the adaptive immune system is the presence of an antigen. An antigen is something that's present on the surface of a pathogen or other foreign substance that will elicit this adaptive immune response. Antigens are often macromolecules such as proteins, polysaccharides, lipids, or nucleic acids. And these are recognized by receptors on the lymphocytes or by our antibodies. While they may seem small to you, antigens are actually pretty big compared to a receptor. And so each antigen has sites called epitopes. And an epitope is a smaller part of the antigen that can be recognized by either an immune cell receptor or an antibody. So while this whole, let's call this a protein here in the middle, is the antigen, different parts of the antigen can be recognized by our immune system, and those little parts are called epitopes. The cells of adaptive immunity are lymphocytes, and these are T cells and B cells. Lymphocytes have some unique properties that are not found in the innate immune system. This includes specificity. That means each clone of T cells or each clone of B cells will be specific for one particular antigen, and more than that, one particular epitope on that antigen. In addition, lymphocytes have diversity. This means we can develop an immune response to just about anything. Another key component of the adaptive immune response is memory. This means once we learn about an antigen, our immune cells remember it so that we can come on strong the next time we see it. Our lymphocytes also are able to determine self from non-self. This is important so that our immune system does not attack our own cells. B lymphocytes, or B cells, are the cells that produce our antibodies, and they mediate our humoral immunity. Whereas T lymphocytes, or T cells, mediate the cellular immunity, and we will talk about several types of T cells. It is important to know that when a particular epitope activates a lymphocyte, whether a T cell or B cell, we make a clone of cells that is specific to that epitope each time. So when you get a particular vaccine, or you get a particular disease and you make an immune response to it, your immune response are clones of T cells and clones of B cells that are specific to the epitope recognized on that pathogen. In order for the adaptive immune system to be activated, we need to have antigen-presenting cells talk to the adaptive immune system so they know what to respond to. So antigen-presenting cells, or you might see them as APCs, will process and then present antigens to our lymphocytes that are involved in the adaptive immune response, so our B cells and T cells. We have three types of antigen-presenting cells. We have our dendritic cells, our macrophages, and B cells. You can see, for example, our dendritic cell. This is a cell that constantly surveys our environment and it looks for foreign invaders. In this figure, this dendritic cell is bringing in a virus through the process of pinocytosis. It processes that virus, and then next we'll talk about how it can present it to an adaptive immune cell. A macrophage works in a similar way through the process of phagocytosis, where it often takes in larger pathogens such as bacteria. It processes them and then presents them to the adaptive immune cell. Our B cells, which are part of the adaptive immune response, can also serve as an antigen-presenting cell. Smaller substances, such as a toxin, may be able to bind to a B cell receptor, which it can then internalize, process, and then present to an adaptive immune cell. So the way our cells talk can include receptors and molecules it secretes like cytokines. And in the adaptive immune response, communication requires a major histocompatibility complex molecule, or MHC. These help our immune system differentiate our molecules from things that may be harmful or foreign. In the adaptive immune system, we have two MHC classes, MHC1 and MHC2. MHC1 is found on all nucleated cells. In the innate immune system, we've talked about how the MHC1 serves as our self-ID for natural killer cells, which survey our cells, asks for identification, and if that identification there, it moves on to the next cell. However, if that MHC class 1 molecule is not there, that's going to activate the natural killer cell to eliminate our own cell. In the adaptive immune response, MHC class 1 molecules also help to stimulate the cytotoxic or cell-mediated T cell immune response. MHC class 2 molecules are only found on antigen-presenting cells. These include our macrophages, our dendritic cells, and B cells. We have a particular set of genes called MHC, or Major Histocompatibility Complex. These genes encode what we call human leukocyte antigens. So what our MHC molecules actually are antigens that define our tissue type. Each one of us has a unique collection of MHC proteins that drives our immune response and can even influence the susceptibility to autoimmune disease. I will note here that there is a third type of MHC molecule called MHC3, and this gene region encodes many of our complement proteins. So we won't be talking about that here in the adaptive immune system. Remember that MHC1 is found on all nucleated cells and our platelets. In the adaptive immune system, the way MHC1 plays a role is by bringing up an antigen in an infected cell and displays it on the cell surface. So you can see here this little black area is the MHC1 molecule and this green triangle is our antigen. Our T cells can then use the T cell receptor to talk to this MHC1 molecule and learn to recognize that antigen. If the T cell finds an antigen on the cell surface that it knows to be this foreign substance, this particular virus it's been trained to hunt, it will then destroy that infected cell. MHC2 molecules are only on those antigen-presenting cells, macrophages, B cells, and dendritic cells. Here, the antigen-presenting cell has already taken in that foreign antigen, processed it, and displayed that epitope on an MHC-2 molecule. This MHC-2 molecule then talks with the T cell receptor, activating a T cell, and then begins that adaptive immune response. Our dendritic cells reside in most of our tissues. These cells continually survey the environment, looking for pathogens and any extracellular material that it doesn't think belongs. In our tissue, they start off as immature cells. These cells are primarily phagocytic in nature. It recognizes those pathogen-associated molecular patterns, or PAMPs, through a pattern recognition receptor like TLR, and that can activate the dendritic cell. It also uses the process of pinocytosis, or cellular drinking, to survey the environment and just sample what's happening in the immediate environment. Once a dendritic cell is activated by a pathogen, it matures and is now an antigen-presenting cell. Now that it's mature, it gets kicked out of the tissue where it is, and it travels on a journey to the lymph nodes. Once in the lymph nodes, the dendritic cell can then communicate with our helper T cells and start to activate the immune response. Dendritic cells are an important bridge between our innate and adaptive immunity. Like our other white blood cells, our lymphocytes come from stem cells in the bone marrow or in a fetus through the liver. In our bone marrow, we have a pluripotent stem cell, which can become any of our blood cells. It then further differentiates into a lymphocyte stem cell. This lymphocyte stem cell has the potential to become a B cell or T cell. If this lymphocyte stem cell becomes a B cell, it stays in the bone marrow to mature. During bone marrow school, B cells get our Ig, or those immunoglobulins that are specific to B cells. And it also gets training on distinguishing self from non-self. Our T cells, if this stem cell is turned into a T cell, will migrate to the thymus for maturation. In the thymus, our T cells go to school just like the B cells, and they learn to distinguish self from non-self. They also are trained to recognize antigens. Thymus school is really hard. If a T cell accidentally identifies self, thinking it's non-self, it gets eliminated from the program. In addition, if a T cell in thymus school doesn't learn to recognize antigens, if it just can't remember, it also gets expelled from the program. So once our B cells and T cells have been trained, they then move to lymph nodes where they wait for activation by our antigen-presenting cells. We have two types of immune cells. We have regulatory cells. These control the immune response. These will be things like our helper T cell, our suppressor T cells, or the antigen-presenting cells. Our effector cells have an effect, so they carry out the attack on the antigen. This would include our cytotoxic T cells, or killer T cells, our B cells that produce antibodies, and our leukocytes, like our macrophages or neutrophils. Our T cells initiate cell-mediated immunity. We have helper T cells that can activate other T cells and our B cells. And we have cytotoxic T cells that can control intracellular viral infections. On our T cells, we have these molecules called cluster of differentiation, or CD molecules. This is how we refer to the different types of T cells. Our CD4 cells are our helper T cells, and these are the primary orchestrators of the immune response. Our CD8 T cells are the cytotoxic T cells, and these destroy infected cells. Our regulatory T cells are identified by the marker of CD25. These can help shut off the immune system when needed. The master regulators of the immune system are the helper T cells. Helper T cells talk to our antigen-presenting cells, and the T cell receptor on a CD4 helper T cell can recognize the MHC2 molecule. It learns about the antigen presented to it by the antigen-presenting cell. It then goes out and releases cytokines to tell other cells about this antigen so that we can develop cytotoxic T cells and B cells. Our regulatory T cells have that CD4 molecule, but they have an additional CD25 molecule. These are important because you need to be able to turn off the immune system. So you can inhibit proliferation of self-reactive lymphocytes or turn off the process when a situation is resolved. These regulatory T cells are still antigen specific, and there's a specific inhibitory cytokine, IL-10, that is secreted to turn off this immune cell growth. The cytotoxic T cells have a CD8 marker on its cell surface. These are often called cytotoxic T lymphocytes. These recognize MHC class I molecules on the surface of a virally infected cell or a cell infected with intracellular bacteria such as tuberculosis, cancer cells, and they're also involved in allergic or hypersensitivity responses. Cytotoxic T cells will destroy an infected cell or a cancer cell through two ways. One, it will release cytokines that are toxic to a cell called cytotoxic enzymes, and it will directly talk to receptors on a cell to initiate the process of apoptosis, or that programmed cell death. Our B lymphocytes regulate humoral immunity. Remember that in our humoral immune system, we eliminate the extracellular microbes and toxins. We also create memory, so we have this heightened response with future exposure. In our humoral immune system, our B cells can differentiate into plasma cells, which are little antibody factories that continue to pump out antibodies that are continually hunting for these extracellular invaders. We also have memory B cells that remember that antigen, so the next time you see this antigen, you can make more antibodies in a really quick way. We have 5 classes of antibodies or immunoglobulins, which are represented by Ig. The most abundant antibody in the body are the IgG. These antibodies circulate in our blood and other body fluid. They constantly bind antigen. It's important to understand that the IgG is the only antibody that crosses the placenta, so it is responsible for the protection of newborns. This is what gets activated when you have that secondary immune response. IgA are found mostly in secretions and on our mucous membranes. So these are things like saliva, breast milk, and our nasal and respiratory secretions, and also on the mucous membranes. IgA antibodies are what protects newborns during breastfeeding. IgM is a big antibody. It has 5 units that clump together in a cluster. You also find IgM in body fluids. These can activate complement. These are the first antibodies that are made in the immune response. So if someone has high levels of IgM, they were recently exposed to an antigen. IgD isn't circulating, but it's found on the surface of the B cells, and this is the B cell receptor. And then our last one is IgE. IgE antibody is found on our mast cells. This antibody is involved in inflammation, and it's the key antibody during the allergic response. And then finally, a quick look at antibody structure. Antibodies have two major areas. There's the variable region and the constant region. The constant region differentiates what type of antibody or immunoglobulin is. Is this an IgG or an IgE? The constant region is the same for each isotype of antibody. So all IgG will have the same constant region. All IgE will have the same constant region. The variable region will be different for every antigen that you have made an immune response to. So we talked about how each B cell clone is going to be specific to one antigen. When we say that, this is represented in the variable region. Throughout the lifespan of your humoral immune response, you're going to do what we call class switching, where you're going to make different constant regions for that antigen. So for example, when you first encounter an antigen and you start that immune response, you're first going to make these IgM antibodies. You will then make a new IgG antibody that has the same variable region, but this constant region is going to be different because it's now going to circulate in your blood, providing your immunity.